Okay, so I'm going to be covering the details of the meat lab, including an explanation on serial dilution. So we started this meat lab with 1 gram of meat. And the idea is to figure out how many bacterial cells were in that 1 gram of meat. If you had to plate this 1 gram straight out on a plate, assuming we liquefy it, there would be just too many cells covering the plate, so we wouldn't be able to know how many, we wouldn't be able to count them. So what we do is we do a serial dilution, and you have first a tube with 9 ml of water, and that's where this gram of meat is put first. So that first dilution is 1/10, because you put 1 gram of meat in 9 ml of water, so total volume is 10. This brings up the total volume to 10 ml, so this results in a dilution of 1/10. We also write it 1/10 or 0.1. Okay, so now we're going to test different dilutions, and so we're going to dilute one more time. We use a pipette for this purpose. We use a pipette, and we pipette out 1 milliliter out of that tube, so that dilution. So this is with pipette number 1. We measure 1 milliliter out, and we're going to pipette that into a bottle that actually is much bigger volume in it. So that's not very proportionate, but that's okay. So in this bottle, we have 99 ml of water. So the final volume is 100 ml. 1 ml in 100 ml final volume is a dilution of 1 to 100, or 1/100ths, or 0.01. And we're going to do one more dilution out of this bottle. We're going to, with a pipette number 2, and again, we use a clean pipette to do that, so we don't carry the remaining cells from pipette number 1 into our third bottle. Our third bottle is also 99 ml of water. 99 ml. So this is an additional 1 to 100 dilution, right? You're taking 1 ml. Actually, let's keep the colors consistent. You're taking 1 ml out of this bottle and diluting it in 99 ml of water. So this is, from here to here, is a 1 to 100 dilution. But really, what we're interested in is how many times we diluted that original gram of meat. So we diluted 10 times. So the final dilution, I'm going to write final dilution over here. Final dilution is, here it's 1 in 10. And for this dilution, we have a 1 to 100, but this is an additional dilution. So here, so this is an additional, it's an additional 1 to 100. So you add that to the 1 to 10 here, you end up with a final dilution of 1 to 1000. And here again, we have a 1 to 100 dilution over here, but that means it's an additional 1 to 100 from what was already a dilution here, and now we're diluting it 100 times more. So the dilution here, 1 to 1000, is diluted 100 times more. So now we're at a dilution of 1 to 100,000. So we diluted 10 times in here, and then we diluted an additional 100 times here. So 10 times and an additional 100 times, that's a dilution, a final dilution of 1,000 times. And then we took some of it out here and diluted 100 times again. So 1 to 1,000 becomes 1 to 100,000. We also use the term dilution factor. I'm going to write like that. We also talk about dilution factor, and this will come up in the equation later. Dilution factor. Some people might like to look at it with a dilution factor instead of just a dilution. A dilution is a fraction, right? It's a 1 ml into 10 in final volume of 10, or 1 ml in final volume of 100. So 1 to 100. But some people like to hear it in the terms of dilution factor. So here we diluted 10 times. Here we diluted 1,000 times. And here we diluted 100,000 times. So the dilution factor is the actual number of times we diluted. Okay, so it's important to understand that we made a first dilution, but then we made additional dilutions. And the important part is to really get back to that 1 gram of meat. How many times did we actually dilute from that 1 gram of meat? Because we need that dilution factor to be able to get back to that number of cells in that 1 gram of meat, which is the original purpose of this procedure. So once you have these bottles of different dilutions, you were asked to plate them into different plates, and we doubled up on the plates. So we had 2 identical plates that were both, so they were labeled A and B, 2 identical plates labeled C and D, E and F, G and H. So really A and B are identical to each other. What they are is we are putting from the bottle, that first 100 ml bottle that has a 1 to 1000, the one that has a 1 to 1000 final dilution. We're going to be plating 1 ml from that, 1 ml. And in the second set of plates, we're going to be plating only 0.1 ml. So much smaller volume. And from the last bottle, we're plating 1 ml in E, 1 ml in F, and 0.1 ml in G, 0.1 ml in H. So each of these pairs are really identical to each other in the sense of there's the same amount plated, so same volume coming from the same bottle. So what you will do at the next lab is you're going to be asked to count the colonies count colonies and then average those numbers. So average the numbers and then calculate your colony forming unit. So let me explain what that is. So the colony forming unit is actually that number that we need to figure out how many cells are in the original 1 gram of meat. So let's remember here, we started with 1 gram of meat, and we are not sure how many bacterial cells are in that gram of meat, right? The meat you buy in the store is never sterile. There's always some bacterial cells in them. The point is not to sell this meat with too many bacterial cells that it could be harmful. So there is a certain number of original cells in here. Original cells, and the idea is to count them. If they're below a specific threshold level, below a specific concentration, then it's harmless. But if there are too many, then this meat is too contaminated and we can't sell it. So the original number of cells, actually, if we were to plate them directly on a plate, we would have too many to count. Too many to count. So we can't really count. It will basically be a carpet of cells. If we try to put that 1 gram of meat in a liquefied form and put it over here, it would just be a carpet of cells, and we couldn't really actually count the number of cells. So that was the purpose of this particular lab, is to do a serial dilution. That's what that's called, a serial dilution. So 2 dilutions in a row, and then that's what we plate. We plate the different dilutions and we count the colonies that appear. So the dilutions allowed us to spread the cells in a larger and larger volume each time. We're spreading the cells, so when we plate them, I can actually see them and count them. So then we count the number of colonies. What's the number of colonies? And when we have that number and we know how much was plated, so this is the volume plated, and we know what the dilution is because that's where we took it out of, the bottle we took it out of. We know that dilution. We plate a specific volume and we count the number of colonies. So when we have all that, we can determine what the CFU number is. CFU stands for colony forming unit. Colony forming unit. You know that one cell, when it lands on a plate, there it is, it's going to actually grow and divide into 2 cells, and then these are going to divide, and then we're going to end up with lots of cells on top of each other, and this is going to form one colony. 1 cell, 1 colony. That's the rule. 1 cell, 1 colony. So if you can separate the colonies from each other, basically counting each colony is actually referring to one original cell. That's the point. So the colony forming unit is referring to the cell, original cell. So it's the number of colonies over volume plated, over volume plated times the dilution. That's the formula.